Welcome back to Lesson 3. At this point, you should have completed all of your data collection and have your data entered into a spreadsheet. Today, we are going to learn how to graph our data and make a scientific presentation of our findings. Let's take a look at where we are in this three-part lesson framework now, and I'll tell you more about what we're going to do today. During the third and final lesson in this series, we'll show you how to graph your data and present your findings. Let's take a look at the diagram of the scientific process we have been referencing to understand how the rest of this series is going to go. Now that we've graphed our data to interpret our findings, the last things we have to do today are share our findings with the rest of the world and come up with some new scientific questions that we may have about this topic. Part of interpreting your findings is making a graph. But let's hold on a second. Why do scientists make graphs? I'd like you to take a moment and write down an answer of why you think scientists make graphs. This should be about 2 sentences, and you can pause this video while you work. Restart the video once you've finished. Nice work. Here you can see some sets of data from a student research project like yours that shows us one of the reasons why scientists make graphs. It can be hard for us to understand whether or not there's a pattern in this data just by looking at the numbers. But if we graph this data, it can be a lot easier for us to understand any patterns in our data and for us to share the results of these graphs in our experiment with the rest of the world. Let's take a look at 2 different types of graphs we could use to share our data. On the left here, we can see an experiment about the number of insects in an area, a forested area or a field area. On the right, we can see an experiment that looked at the distance from water and the number of plants in an area at each distance from water. Here we can see a bar graph and a scatter plot. These are the 2 main types of graphs you might use to graph your data. As you can see, it's pretty easy to tell that in the bar graph, the field area had more insects than the forested area. On the scatter plot, we can pretty easily see that near the water, we found more plants, and then as we got further away from the water, the line decreased, showing that we found fewer plants. In the next segment, we're going to show you how to make graphs and analyze your data. A full guide for how to do this can be found linked in the description of this video. I'm going to demonstrate how to make a graph using data from my research project. But before we go into that, let me show you how I collected that data. My research question was, how does habitat affect the number of insects in an area? My hypothesis was that there would be more insects closer to the river where there is food and water for them. How I did my experiment was I used something called a transect. I laid out a tape measure and walked in a straight line starting at the river, across the near river area, a meadow, into the forest, and collected insects along the way. I stopped every 2 meters and collected insects 4 different times and continued in a straight line until I was all the way into the forest. So here is the data that I collected. I started at the river, which was 0 meters, and I went 2 meters away through the near river area, through the meadow area, and into the forest. Along the way, I recorded the types of plants that I saw where I was catching insects, as well as the number of insects that I caught in each trial. As you can see, I collected insects 4 times at each location. In order to graph this data, we're going to have to organize it a little bit so that we can make the graphs that we want. A great place to start here is to calculate the average. Here I have the average number of insects that I collected per each trial. If you don't remember how to calculate an average, remember that you add up all of the numbers and then divide by the number of numbers. Here I did 4 trials, so I divide by 4. I did this for each one of my experiments. The cool thing about my data here is that I could use either a bar graph or a scatter plot to calculate what I've done. Let's take a look at using a scatter plot first. I have this set up in a different tab so I don't mess up my original data. You can see that here. I have the same data that I had before, but over here I took 2 columns and put the distance from water and the average number of insects per trial. If I compare how the number of insects per trial changes with distance from water, that might help me understand if there's a pattern there. To make a graph out of this, I simply select all the numbers and headers in these cells and go over to Insert and choose Chart. I did this over here already, and I know we're going through how to make these graphs pretty quickly, but you can always use the stats and graphing guide that's linked in the video description. Here you can see a pretty good pattern where we saw more insects near the water and fewer insects further from the water. But as you can see, there's a little bit of variability, and it wasn't always a linear increase. Next, I can show you how we could make a bar graph of this data. To use a bar graph, I'm going to be looking at these 3 different habitats that I collected insects in across my transect: the near river, the meadow, and the forest. I have that organized here. What I want to do is take an average of the number of insects in the near river places and compare that to the average of insects in the meadow places, and then compare that to the average of the insects in the forest places. To do this, I'll calculate an average. One nice thing about doing this in Google Sheets or Excel is that the computer has some programs that can help you out. I can type the command equals and then the word average and select the numbers I want to average. You can see here it's near river, so I can select the insects in the near river area and hit enter. Cool, the average number of insects I collected in the near river sites is 26.2. I'll do the same for the meadow and the forest now. Perfect, the meadow was actually a little bit higher. And then for the forest, wow, look at that. The forest was a little bit lower. It's interesting to see how when we graph our data in a slightly different way, we might see slightly different results. To make a graph, I'll do the same thing here of select the data, go to Insert, Chart, and I want this to be a scatter bar graph, and so I'll change this to a bar graph like that. I like to also put data labels on here so I can see the patterns more easily. There's more detail on how to do this in the Graphing and Stats guide linked in the video description. A neat thing here is it's pretty neat to see that there wasn't much of a difference between the near river and the meadow sites, but the forest had a lot fewer insects per trial. You can pause the video now while you make your graphs and restart once you're done. You can reference the stats guide in the description of this video or rewind parts of this video if you need to see it again. Restart your video once you've completed making your graphs. Nice job! Now that you've made your graphs, we can start to investigate what patterns are there in your data. When we look at a graph, it's important to start with the broadest patterns that we can see before getting too specific. If we look at the examples from my graphs, we can see that the scatter plot, that we find more insects near the water and fewer insects further from the water. That's the main pattern that we can see there. In the bar graph, we can see that we pretty much found the same number of insects in the near river and meadow areas, but we found a lot fewer insects in the forested area. The second thing we want to think about when we look at our data is how strong are the patterns that we're seeing. For example, the difference between the number of insects in the near river site and the meadow site is really small. Even though there are more insects on average in the meadow, that difference is really small and we saw some variation in our data. I probably wouldn't be confident that one was higher than the other if I was going back to measure these again. When we're thinking about how to interpret a scatter plot, there are a few different types of things we want to look at. The first is the line slanting or is it pretty flat? A flat line suggests that as one of your variables changes, the other one stays the same. The other thing to look at in a scatter plot is how close are the points on your graph to the line of best fit that is running across your graph. If all the points are really near your graph, you can be more confident in your results. If many of the points aren't close to the graph, then you might be less confident in your results. To understand this, let's take a look at this scatter plot side by side with some other data I collected in the same place. Here we can see the same graph of distance from water and number of insects. On the right, we see a new graph that I collected in the same place. This was the distance from water and the number of different types of plants I saw where I collected my insect data. As you can see, the trend line of these two graphs is pretty similar, but the graph on the right with the number of plants, the points are much further from the line, whereas with the graph on the left showing insects, the points are much closer. The pattern in the graph on the left about insects is a lot stronger because the points are closer to the line, whereas the pattern in the graph on the right is a lot weaker. It might not be something I would be able to observe if I went back and did the same experiment again. I would like you to pause this video now and try to answer these two questions on this slide. What are the main patterns in your data? And how strong are those patterns? Be sure to describe the strength of every pattern you can see in your graph. This next section is for high school students. If you're in middle school, you can skip this part because it involves some more complex math, but you're welcome to watch along if you'd like. Up until this point, we've described the strength of our pattern by just looking at the data and kind of approximating what that means. A more advanced and accurate way to do this is to use mathematics and a statistical test to calculate exactly how strong the pattern in your data is. We use different types of statistical tests depending on the graph that you would be doing. I won't go into the details of how to do these here, but you can find full instructions on how to do so in the stats guide that's linked in the description of this video that we've been talking about already. For a scatter plot, we use the statistical test called a linear regression, which gives us an r-squared value. For a bar graph, we use something called a t-test, which gives us a p-value that we can use to interpret our results. I have the results of the statistical tests for the graphs on this page written up there, and they mostly support what we talked about so far. That the patterns in the number of insects versus distance from water was a lot stronger than the number of plants versus distance from water. Also, that there wasn't really a meaningful difference between the number of insects in the near river and the meadow area, but there was a meaningful difference between the number of insects found in the near river and forested area. Let's take a look at the scientific process here to understand how we're going to finish out today's lesson. Now that you've completed your data collection, it's time for us to interpret our findings so that we can share the conclusions that we've come up with and maybe come up with some new research questions that we have to help us investigate this topic even further. The key parts of a good scientific presentation is that you describe how you did your experiment, that you share your conclusions, and that you give the audience some ideas as to how your experiment could be improved or other research questions that you have. All of these three things are really important because a key idea of science is that a different scientist could do the same experiment in a different place and get the same results. This is called repeatability. In addition to repeatability, another important part of science is that scientists share their findings with one another so that we can all benefit from the discoveries you have made. In this next section, I'm going to walk you through the parts of a scientific presentation so that you can make one on your own. On the first page is your title. This is a short 5 to 6 word description of your project that also includes your names and your affiliations, such as your school or your science teacher. The next page is your introduction. It should include some general information about the subject of your research. Here you want to let your audience know why your research is important and tell them when and where you did your research. It's important to give your audience a good context for your project so they can understand how and why you did it. On the next slide, you'll tell your audience what your research question was and what your hypothesis is. After that, you will show them how you did your experiment. In your materials section, include the scientific materials you used to do your experiment, and then describe how you did your experiment with the method section. Here you want to use just enough information so that another scientist could do the same experiment. Bullet points work great here. In the evidence slides is where you're going to put your graphs. You can copy and paste your graphs from Google Sheets into here. Remember that every graph should have both the horizontal and vertical axis labeled, as well as a one-sentence caption describing the main patterns in the graph. It works best to only have one graph on each slide. If you have multiple graphs, make multiple evidence slides. The discussion slide is one of the most important ones of your presentation. This is where you'll tell your audience what your results mean. Be sure to describe the main patterns in your data, how strong those patterns were, how do your results compare to the hypothesis or prediction that you had. Also include some analysis of your findings. Why do you think your experiment had the results you observed? And how could your findings be helpful to other scientists who studied this similar topic? Just like the evidence section, if your discussion section is getting a little long, you can always split it and put it onto 2 slides. The last slide in your research presentation is about future research. This basically asks you the question of how could you improve your experiment to better test your question, as well as now that you've done your experiment, what new questions do you have? This reflection is a really important part of the scientific process. It's important to be honest about where you could improve or other ways you could find an answer to your research question. Your homework for today's lesson is to finish your scientific presentation and then to complete the scientific process by sharing your work. You can share it with your teacher, your parents, your friends, or me, Spencer at headwatersscienceinstitute.org. Check out these video clips of students like you sharing their scientific presentation with their peers.
We conducted our experiment on October 2nd and October 8th during low tide in the Rodeo Lagoon watershed, and we wanted to determine if there was a correlation between salinity and plant vegetation, so plant coverage on different locations along the watershed. And we measured the salinity, temperature, dissolved oxygen, plant coverage, and type along these locations.
Our research question was, how does the salinity of different areas affect the vegetation? Our independent variable was the distance from ocean, and our dependent variables were salinity, temperature, plants, and dissolved oxygen.
So as you can see from the first graph, water temperature was slightly higher in locations closer to the ocean, and in the second graph there, salinity decreased as you moved further from the ocean.
Nice work, everyone. You've done scientific research, and that makes you a scientist. Not only are you an expert in the topic that you studied, you've also learned the skills of how to do scientific research. This means that the next time you have a question you can't find an answer to, you know how to go outside and create an experiment that can help you answer that question. You can look for more videos like this from Headwaters coming soon, where we'll show you really cool systems that you can create your own research project in. That's all for now, and we'll see you next time.